Assalamu alaikum. In this lecture, we'll be talking about polymerase chain reaction, PCR. You've heard of PCR in the news in the past 2-3 years during the COVID-19 pandemic. And PCR has revolutionized the field of molecular biology, molecular medicine, and even molecular archaeology. So it has done tremendous improvement in the way we know and analyze DNA. So there were several challenges when it came to studying DNA. Some of these challenges were related to genetic variations. So we're talking about the existence of short tandem repeats. We're talking about variable number of tandem repeats. We're talking about variations such as single nucleotide polymorphisms and the presence of mutations. How could we identify these things, and how could we take advantage of genetic variations as well? So a huge challenge was actually also how can we handle minute amounts of genetic material, that is small amounts of genetic materials. For example, in bones, old ancient bones and skeletons found here or there of dinosaurs, or even ancient humans, early human beings, or even a hair in a crime scene, or a blood spot, or whatever. How can we identify organisms, whether whether identified existing known infectious agents or novel infectious agents, such as the SARS-CoV-2, and I don't know what's coming next. So how can we identify these things? How can we study these things as well? And the invention of PCR, as I said, has revolutionized everything related to DNA and nucleic acids in general. So this technology was invented by a scientist known as Kary Mullis in the 1980s, and it allows basically the amplification, and this is the point of this technology, the amplification of a piece of DNA or a region of it. So we can amplify this whole thing, or we can identify a little region or big region of this nucleic acid. So we can amplify billion folds. Okay, this is really huge. So it is really extremely sensitive. It can detect even a single DNA molecule in a sample, and it can amplify it. Of course, the inventor got the Nobel Prize for his invention. Now, the piece of DNA, by the way, that can be amplified. It can be linear. It can be circular, but the final product is linear DNA. So it is a reaction. Okay, so it's a biochemical enzymatic reaction. And it is a chain reaction, meaning that it is a consecutive reaction. So mutasalsal. So basically it goes, the same reaction is repeated several times. Since it is an enzymatic reaction, it requires an enzyme, and the enzyme used is DNA polymerase, a special type of DNA polymerase, and we'll talk about it in a second. So since it is an enzyme, it requires substrates, and the substrates are deoxyribonucleoside triphosphates, okay, because energy is needed. So we're talking about deoxy A, deoxy G, deoxy C, and deoxy T. Okay, now it requires a template, and as I said, the template can be circular, it can be linear, and of course the DNA polymerase requires a point where it can start amplifying DNA or synthesizing DNA, and so we use a pair of primers. And the primers are known as primer 1, primer 2, or forward primer and reverse primer. Now, so we have the template right here, and we want to amplify this region. So we use the forward primer, the reverse primer, and they surround the region that we want to amplify. Okay, so you have synthesis going on from the 5' end to the 3' end. So there's directionality, 5' to 3'. It's anti-parallel, of course, and the newly synthesized DNA contains the one strand that is old, the parental strand, and the other strand is the newly synthesized strand. Okay. So each PCR reaction goes into cycles, and these cycles are repeated. So you have cycle 1, cycle 2, cycle 3, cycle 4, and so on. So each cycle consists of 3 stages or 3 phases, and these are known as 1, the denaturation stage or step. We have the annealing step, and we have the polymerization or the DNA synthesis step. So the denaturation step occurs at 95 degrees. At this temperature, the 2 strands are separated from each other. Then we go into the annealing step. Now in this step, the primers anneal or they hybridize to the DNA. So we have binding to the forward primer, the reverse primer, they bind to, each one binds specifically to its strand. Again, the primers surround the region to be amplified. Then the temperature goes up to about 72 degrees. This is the optimal temperature for the DNA polymerase. Okay, so we have 95 degrees, then it goes down to about 50 to 60 degrees. Well, it can go up to 70 degrees. It depends on the primers and their annealing temperature, the temperature that is optimal for their hybridization to the DNA. And then we have a temperature of 72 degrees at this in the third step for DNA polymerization. So notice that the temperatures that we're talking about are really high temperatures. We're talking about a temperature of about 95 degrees. We're talking about a temperature of 70 degrees and so on. So these temperatures cannot be handled by the human DNA polymerase. So we have to use a special DNA polymerase. And the DNA polymerase that we use was isolated from a bacterium known as Thermus aquaticus. Now, thermos for temperature, okay, thermometer. Aqua means water. So Thermus aquaticus was isolated from hot springs in the US, in the Yellowstone Park, and this is how it looks like. So they can handle really high temperatures. Of course, you can just imagine that we can also isolate bacteria from Hammamat Ma'in springs, beautiful springs in Jordan. So, you know, I encourage you to go and visit them, relax during the, you know, break between semesters. So I'm promoting tourism in Jordan. So we use DNA polymerase from these bacteria. So Tac polymerase. So the polymerase is known as the Tac polymerase from Thermus aquaticus. And this is how the reaction goes. So in the first cycle, we have denaturation taking place, separation of the 2 DNA strands. Then we have the annealing step. Here, at a certain temperature, we have the primers, the forward and reverse primers sitting, hybridizing, annealing to their complementary sites, and they must be, again, they must surround the region that we want to amplify. And then we have the temperature going up again to about 72 degrees. This is the optimal temperature for the for DNA synthesis by the DNA polymerase, the Tac DNA polymerase. Now notice that we have the product extending to the left and right. So this is not really the the product that we're looking for. Now in the second stage, we have synthesis going on as well from these two template molecules. So we have synthesis again for the 4 strands, and we have again we have amplification of DNA, but we don't have the final product. Now in the third cycle, we have synthesis going on again, and at this point we have our product, and this product is then amplified at cycles 4 and 5 and 6 and so on. And please watch the animation video that I will post on your site. Okay, so here we have amplification of DNA. So we have the reactions go on for about up to 30 cycles. We can go up to 35 cycles if we want. Anyhow, so at the end we would have about 250 million amplicons or amplified DNA fragments. So each cycle presumably doubles the amount of DNA. At the end of the reaction, we take our samples and we analyze them by gel electrophoresis. So here we have the standard marker. We know the sizes of these DNA bands, and these are our samples. And you can see that what PCR does for us is that it specifically amplifies a certain region, and we only have one band, meaning that we have lots of DNA right here in this band right here. We take small amount of our sample at the end. So you can amplify specifically a certain region, and you get a pure DNA at the end. That's beautiful. The step that is really important is the annealing temperature. This is the step where you have the primers binding or annealing or hybridizing to the regions that it should be complementary to it. So if you remember, we talked about DNA with the melting curve and the melting temperature, which is where you have double-stranded DNA, and as you increase the temperature, the DNA, the hydrogen bonds between the two DNA strands get broken. So the higher the temperature, the more bonds that get broken, hydrogen bonds. So eventually you get 50% of the DNA bound or hydrogen bonded, and 50% is not. So this is the melting temperature. Now as you go higher in temperature, the DNA becomes single-stranded. So we talked about the factors that affect or influence this DNA denaturation and the melting temperature, and we talked about the composition of DNA of A and T versus G and C. So the more G's and C's, the higher the melting temperature. Okay, and we also talked about things like salts and ions and stuff like that. So anyhow, if you have an annealing temperature that is low, this allows for non-specific hybridization between the primer and the DNA strand, the template, to take place. And that results in non-specific amplification. Remember that we have regions in our genome that are homologous to each other. That is, they have the same sequence. So yes, you can have non-specific amplification, and this is shown in these two figures right here. So this is amplification at different where you have different annealing temperatures. So as you can see, the lower the temperature, the more non-specific hybridized amplification taking place. At a certain temperature, now you have specific amplification, a single band, as you can see in these figures right here. So the higher the annealing temperature, the more non-specific, the more specific amplification that would take place. Now, what would happen if you have really high temperature? In this case, nothing would happen, of course, because there would be no hybridization between the primers and the DNA template.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Tac polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Tac polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Tac polymerase functions optimally at this temperature and begins polymerization, adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA. The PCR reaction mixture contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of cycle 25, there are more than 33 million copies of this double-stranded target region.
Okay, so quantitative PCR, basically the basic qPCR method utilizes a chemical known as Sybr green. Now, Sybr green is a chemical that binds to double-stranded DNA. Now, when it binds to double-stranded DNA, it fluoresces. Okay, so the more double-stranded DNA you have in a sample, the stronger the fluorescence, and this fluorescence can be detected by an instrument. So again, in PCR, DNA is amplified. So at the end of the cycle, when the DNA is renatured, you have Sybr green bound to this double-stranded DNA, and since it's amplified, you have at the end of each cycle, you have more and more and more fluorescence. Okay, so the idea here is that if we have more of DNA to start with, you will have the fluorescence, the signal detected early on. Okay, so let's say that here we have we start with, so the idea is that let's say that you have 2 individuals and one of them has severe symptoms versus the other, and you expect that the one with these severe symptoms has a higher bacterial load or viral load. Okay, so we take a sample from this individual and that individual, and we extract DNA. We isolate DNA from the sample and we amplify it. And we use specific primers for the bacterial DNA or the viral DNA, so that only bacterial DNA would be amplified or viral DNA would be amplified. So primers are really important, and the temperature as well, annealing temperature. So let's say here we start with 1 million DNA molecules. And here we have 100,000, here we have 10,000, here we have 1,000, 100 DNA molecules, and here we have 10 DNA molecules. And then we have a sample that does not have any DNA whatsoever, just water. Okay, so we amplify DNA. Now, the sample that contains 1 million DNA molecules, you have the signal. So this is cycles, x-axis cycles, and here we have the intensity of the signal. Okay, so here the sample with the 1 million DNA molecules, the signal is detected at cycle 10. Well, why not earlier? Because that's the limitation of the instrument. It cannot detect a signal that is low. Okay, but so you have the signal detected at cycle 10. Now, the sample with the 100,000 DNA molecules, you have the signal detected at cycle, I don't know, let's say 14. Now, the sample that contains 10,000, you have the signal detected early on at cycle, let's say 17, and so on. So the lower the DNA, the starting material is, the starting DNA concentration is, the later the signal would be detected. And that gives you an indication for how much starting material you have in a sample. Now, how about the sample with no DNA? Notice that there is no signal whatsoever that is detected, and that's a good thing. Now, we place a line, and it's arbitrary. It's like it's of our choice. It's not, there's nothing sort of like scientific about it. We add a line. This is a threshold line. Okay, العتبة. Okay, threshold is like عتبة. So, and we place this line, and it should be above the negative, the signal of the negative sample, of course, and that tells us where the signal is detected, and this is known as the CT. I'm sure you've heard about CT in the street, because everyone talks about what's your CT, what's your CT, you know, when detecting the coronavirus. So we have the CT right here. This is the threshold cycle. The cycle where the signal is detected, and it's different between or among the different samples, right? So the higher the starting material is, the earlier the signal is detected. So now, with the Sybr green, you don't know what's really amplified. You hope that your DNA is amplified. Okay, your target DNA is amplified. But you can get non-specific amplification. Well, how? And Sybr green does not differentiate between this or that, the specific DNA or the non-specific DNA amplification, because it binds to double-stranded DNA. So how can we be sure that we're amplifying our DNA? Well, we can at the end do gel electrophoresis, and we can see where the bands are. If we have a single band where we expect it to be, then that's great. But the instrument itself can do this by using melting curve analysis. Again, it depends on the melting point, the point where 50% of the DNA is denatured. And what a melting curve does for us is that it tells us the change of fluorescence observed when double-stranded DNA with incorporated Sybr green dissociates or melds, you know, quote unquote, melds into single-stranded DNA as the temperature is raised. So here what we have is, again, the instrument detects change in the fluorescence. So we, at the end of the reaction, we increase the temperature, and the DNA becomes single-stranded, right? So it gets denatured. If we have a single product, then we should have a single peak right here. This is the highest change of fluorescence as the temperature is raised. So if we have a single peak, that means that we have a single product. If we have two or more peaks, that tells us that there are two or more products, because each one of them will have a different sequence or different composition of A's and T's, and that means that they should have different melting temperatures. Well, again, Sybr green based qPCR is not really that specific, and it's limited. So let's talk about another method that is more specific, it's more sensitive, it's more reproducible. That is, every time we do it, we get the same results, and it allows multiplexing. I'll talk about multiplexing in a second. So it's known as Tac Man Qpcr. Now, in addition to the primers, we also use a probe. Now, what's special about the probe is that, one, it's specific to the target that we amplify. That's one. Two, it has a reporter and a quencher attached to it. Now, the reporter is basically a chemical that fluoresces. It gives a fluorescence. The quencher is a suppressor of the reporter. It suppresses the fluorescence that can be emitted from the reporter if it is close to it. So if the quencher is close to the reporter, that is as close as this, being attached to the same probe, now the reporter does not fluoresce. Now, if they get separated, then the reporter can fluoresce. So, we use a special Tac polymerase. This Tac polymerase has a 5' to 3' exonuclease activity in addition to the polymerase activity. So, as it extends the DNA, as it synthesizes the DNA, once it reaches the probe, it activates the exonuclease activity. So it starts degrading, cleaving the phosphodiester bonds between the nucleotides of the probe, releasing the reporter, and now it gets away from the quencher. Now it fluoresces. So we add a lot of probe, and more and more and more of the probe molecules would become degraded by the Tac polymerase. So you get, at the end of each cycle, you get more and more and more signal coming out. So it's more specific because the probe is specific to the target DNA that is amplified. Okay, so if you have amplification of another target that is not specific, the probe does not bind to it, and as a result there would be no fluorescence. It's more sensitive because the signal that is emitted from the reporter is stronger than the signal that is emitted from the Sybr green. It's more reproducible because you would get the same results over and over again, because not only that we're using primers that are specific to the target that we want to amplify, we're also using a probe that is specific to it as well. And it allows for multiplexing. What does that mean? It means that you can have a probe, you can amplify different targets. You can use different sets of primers in the same exact reaction. So you can have a tube that contains the same template. You have a Tac polymerase. You have the same deoxyribonucleotides, but you also have different primers, different sets of primers that can amplify different regions in the template, and different probe molecules that would bind to this amplified region and this amplified DNA and that amplified DNA, and each one of them would have each probe would have a different reporter emitting different signals. So you have a red signal, and you have a green signal, and you have a yellow signal, and you have a blue signal to each one of these probes. So you can amplify different regions, and this is shown in here, for example. So right here, you can have a signal emitted for this bacterial DNA, or this DNA of this specific bacteria. You have a signal emitted from another gene for a different bacterial species, and you have a third one and a fourth one. So let's say that you are a geologist, for example, or a geological microbiologist, and you want to identify the different types of bacteria that exist in a a sample of sand or whatever. So you take it and you want to detect different bacterial species in that sample. So you use different sets of primers and you use different probes for each one specific for specific bacterial species. So you amplify it in one single reaction. You have amplification of 4 different regions in 4 different bacterial species, 4 different genomic DNA molecules, and you can detect them. And you can also do sort of like quantification as well, because you have signals emitted at different threshold cycles. And that tells you something about how much of this bacteria you have in this sample or that sample and so on. And this actually was done in the coronavirus, except that the coronavirus, the genetic material is RNA and DNA. We'll talk about that in the next lecture. But you can amplify different regions in the SARS-CoV-2 quote-unquote DNA in a single sample to confirm that this sample contains the SARS-CoV-2. So this is multiplexing. Beautiful technology. Allah ma'akum.